lives run on our brain's incredible capacity for memory. We spend so much of our lives trying to form them.
A loaf of bread, a container of milk, and a stick of butter. Sir, could I have a loaf of bread, a container of milk, and I can't remember.
And accessing them, and it makes you wonder, why do some things seem to stick while some fall right out? Do you want to remember? Sure, the cortex might store memory long term, and the mesolimbic system really pulls through when there's a reward at the end, but if you want to use it, you won't get anywhere without us. Short term or long term, emotional or sensory, spatial or procedural, and so much more. All here at the hippocampus. Free with your brain subscription. There are no neurons for your birthday in 2022 or triangles. Instead, memories are encoded by connection between neurons known as the synapse and the greater network several synaptic connections make. On one end of the synapse, we have axons that send messages out and dendrites, which receive axonal messages. Say you see a fuzzy animal creeping around your house and someone calls it a cat. Neurons from visual and auditory cortex each project to the entorhinal cortex, which projects the two different neuron networks in the hippocampus. The visual memory neurons and the auditory cat memory neurons will fire with a change of voltage called action potential in response. This drives the release of glutamate, a neurotransmitter, which both networks receive. And neurotransmitters are molecules that make the message more specific to cells that carry that receptor. There are two relevant receptors that are glutaminergic in the hippocampus, but the ones most involved in memory formation and recall are N-methyl D-aspartame receptors, aka NMDAR, and alpha-amino 3-hydroxy-5-methyl 4-isoxazole propionic receptors. We're calling it AMPA. Now, you may be thinking, it's great that these neurotransmitters get passed around, but how do they actually keep memory long-term? That's a great question. The reason lies in what's known as Hebbian theory. In short, cells that fire together, wire together. When axons release glutamate towards another cell's AMPA receptors, the cell experiences an active potential. The change kicks off a magnesium ion from the pore like a cork, and the NMDAR is open. AMPAR is important, but NMDAR is what really runs the show. Once NMDAR fires, this neuron becomes more sensitive to the other neuron's activity, and it starts an intracellular cascade that strengthens synapses and adds more AMPAR to the cell membrane. Next time you see a cat, the other network is now even more sensitive to this glutamate released and fires an association, so you think cat without needing to be told. This synchronous firing process is called long-term potentiation, and as they keep firing together, they'll keep wiring together. The cool thing about the hippocampus is that this firing isn't just created by experience repetition, but recall. So even as memories are stored in other parts of the brain like the cortex, the hippocampus is like a way station where these networks of synapses between several cells must reactivate so you can remember. The more frequently this happens, the more you strengthen these synapses and the faster and easier recall becomes. But without this strengthening, synapses experience pruning to make space for more relevant memories. So the next time you're looking for a carton of milk, a loaf of bread, and a stick of butter, remember, you gotta use it or lose it.